Every day, 30 million metric tons of carbon dioxide dissolves into the world's ocean. This is changing the chemistry of seawater and threatening marine ecosystems. Today, we'll recreate this process in our very own UWorld lab and see how various water samples are affected by carbon dioxide. This topic is a major part of your AP Environmental Science course, so stay tuned to learn the science behind it. So what is the science behind carbon dioxide affecting oceans? How exactly does carbon dioxide enter our oceans and impact pH? Well, close your eyes for a second and imagine yourself on a beach. What do you hear? What do you feel? In my mind, it's a sunny, warmy day. I feel the breeze and hear the waves crashing along the shorelines. Okay, open your eyes. Bye-bye beach. I'm gonna miss you, but I'll be back soon. The mixing associated with wind and waves, as I was picturing it, is one of the biggest reasons why atmospheric CO2 enters and dissolves into oceans through the process called gas exchange. Another key reason for CO2 moving into oceans is diffusion. Diffusion occurs when molecules move from an area of high concentration to an area of low concentration. Take it back for a second and imagine when your mom told you to clean all of your stuff from your room. What did you do? Neatly put it away where it belonged? No, I know I didn't. I used to just stuff my things into the closet as much as I could and then quickly close the door. I created a concentration gradient doing that. But as soon as I opened the door, all of the stuff would just fall right back on out, right? That is moving from an area of high concentration gradient to an area of low concentration gradient. Similarly, there is so much CO2 in the atmosphere, it naturally diffuses or moves down its concentration gradient and enters oceans. Once CO2 enters the ocean, it undergoes a chemical reaction that forms carbonic acid. This acid then releases hydrogen ions, which lowers the water's pH. In addition, these hydrogen ions reduce the availability of carbonate ion, a crucial molecule used by coral and shellfish to build their skeletons and shell, making it harder for them to survive when the pH of oceans decrease. In this lab, we'll measure changes in pH in various water samples exposed to CO2. Then at the end, we'll add some sodium bicarbonate, a chemical that dissolves to create buffers, which will increase the initial pH as well as buffer the solution, which should limit changes in pH. So here's what we'll need to do this experiment. We'll need some flasks with stopper and tubing. On this, I've set it up so we have an air stone on the end of it, and that'll become important in a second. We need a couple beakers. I have one here, and I have another one here, and then I have some more tubing with another air stone. This tubing is going to get connected to our air pump that we already have plugged in and ready to go. We'll need our water. Obviously, we need a lot of water. So here is some distilled water. We also have our salt water, and this salt water is going to mimic our ocean water. I prepared this earlier, which is why it's in a giant bucket. Next, we'll need some effervescent tablets. These tablets dissolve in water and serve as our source of carbon dioxide. Next, we'll need a pH meter. This pH meter will continuously kind of test the pH of our water as it changes throughout the experiment. We will need a stopwatch. And finally, we'll need some sodium bicarbonate, which I've already weighed out into 1 gram increments. So the first thing we need to do with all of this is set up our control apparatus. So in this first setup, all we need is our beaker, and we'll add about 200 mLs of distilled water. And I'm using distilled water just to make sure that there is no ions or any buffering capabilities within that. Once you have that, you take your tubing with the air stone and you're going to connect that to your air pump. This will be used as a control because instead of pumping carbon dioxide into our distilled water, we're just pumping natural air. So with this first experiment, what we're gonna do is we are gonna test the pH of it using our pH meter. And we are gonna take a pH reading for about 2 total minutes to see how regular air changes it. So once you have your pH meter submerged in the liquid, wait a couple seconds until your reading kind of flattens out. And then using the stopwatch, you can kind of time that. And our pH is fluctuating a little bit, but it really hasn't changed. It's about 7.04. So once we're at the minute mark, we are gonna remove our air stone from our solution, and we will continue to measure the pH for another minute. So we're just about at a minute. We'll go ahead and pull our air stone out, and we'll continue measuring the pH of that. So at about a minute 30, we'll take another reading. I just took it. It's still around 7. And lastly, now that we're done at our 2-minute mark, we can take it out. And I looked at the pH right before I took it out, and we can start recording our data. We started with pH of 7.1. And then after about a minute, we went to 7.04. And then we ended at about a pH of 7.0. So it changed, but it didn't change really all that much with our air stone. And again, because we have distilled water and this is our air stone, this will be kind of our control. So now let's test and see what it looks like with the creation of carbon dioxide. For the second part of the lab, we are going to have a little bit more intense setup. This time, we need our apparatus with our tubing and our air stone attached. And you can see I've already attached that. The stopper has that tubing. So for this, we need to fill our flask with about 500 ml of water. And then we will add about 200 mLs of distilled water to our beaker. For this, you want to make sure you have everything ready before you drop your effervescent tablet into your flask. So we have our stopwatch reset. Everything is ready to go. Our pH meter is on. We are going to drop this effervescent tablet into the flask and quickly cover it. We'll measure the pH again for about a minute, and then we will take out the samples. We started of a pH at 7.64. And we're getting close to our 30 seconds here. And you can see all this carbon dioxide is being created is getting pushed into this water. At 30 seconds, we have a pH of 6.09. And it's quickly dropping. So now the bubbles have died down quite a bit, but there's still some happening. Our pH is continuing to drop. And so about rounding off our minute here, the pH is 4.2. Now we're going to take our air stone out and continue measuring the pH for another minute or so. So at a minute 30, we're at pH 4. And so now we have a pH of 3.96. In this sample, we used distilled water and we added our effervescent tablet to mimic the production of CO2. And our pH started at about 7.64, then went to 6.09. After a minute, it was 4.2, which is a huge difference from what we had here. And then after that other minute, we ended at 3.96. So after we took the air stone out, there really was not a huge change. But you can see how much of a change we had with our effervescence. And this change was due to the production of CO2. And similarly to our oceans, that CO2 that's produced in here is transferred through the tubing, kind of like the atmosphere, and then is exchanged with our water. So now that we have our baselines using distilled water, we can repeat these same experiments using our salt water. And this is the salt water I created earlier, and it's going to mimic the ocean water. So any changes that we see theoretically in our lab will be similar to those that happen out into the real world. So stay tuned while I clean this up and set up the next experiments. Okay, now that we're all set up, we can start doing the experiment with our salt water. So similarly to the distilled water, we're going to start with our control. And I remember that control is the air pump pushing water in our salt water. So same thing, air stone, pH for about 2 minutes. We're starting at a pH of, let's stabilize, 7.8. And after 30 seconds, we're still at 7.8 for pH. So again, not anything is changing with our air stone. And then we're about 7.86 after a minute, and we're gonna take this out and we're gonna measure the pH for another minute or so. And at a minute 30, there's no change in pH. And again, here at about 2 minutes, we're still at 7.84. Not a whole lot of change happened with our air stone, which is what is expected. What did happen though is our pH is a little bit higher for our ocean water. And the pH of our salt water with our air stone started at 7.8. Then after a minute, it was about 7.86. And then after another minute or so, we had 7.84. So overall, not a lot of change with our air stone, which was expected since our distilled water showed similar results. So now comes the fun part, is we set up our apparatus, but this time we're going to use our effervescent tablet to mimic our change in atmospheric carbon dioxide. Then you need to make sure you have all of your tools ready to start measuring. We have our pH meter, we have our stopwatch, and we have our effervescence. So let's go ahead and add our air stone to our salt water, and goes our effervescence. So we started with a pH of about 7 or 8.4, excuse me, and it is quickly dropping. After 30 seconds, we're about 7.78. So it's dropping, but it's not dropping as quickly as our distilled water, because remember, our ocean water has a bunch of dissolved salts in it, which buffer the change in CO2. So after just about a minute, our pH is starting to stabilize and we're at 7.45. So now we're going to take our air stone out and measure for another minute. At a minute 30, we are at 7.3. So after that final reading, we're at 7.27. And our starting pH for this one was around 8.4, and then it went to about 7.78, and then about 7.45 when we took out our CO2, and essentially our last minute then went to a final reading of 7.27. And throughout these, I'm doing a little bit of different skipping around on our readings. The main thing that's important is our initial, our middle, and our final reading, right? So if we're getting one with a little bit more data here, in this case, you can just use one of these right here. This will count the same as our middle one in our initial control reading. So now we have some interesting data, right? We can start seeing some changes. The big thing we can compare is our distilled water with CO2 with our ocean water with CO2. And remember, I talked about our ocean water having some salts already dissolved in it. And you can see the effects of those salts. We had a huge change in pH with just our water that was distilled. With our salt water, yes, it decreased, the ocean acidified, but that change is much slower than what happened with our distilled water, again, because we have all those salts in it. So now for the last experiment, let's see what adding 1 gram of sodium bicarbonate into our water does. And so stay tuned while I change this out and we get ready to repeat this experiment. So now that we're all set up, we can continue with the last part of our experiment. And that last part is to add 1 gram of sodium bicarbonate into our various waters. So like I said earlier, I've already weighed this out. We have set up our distilled water here and our salt water back here. So we'll go ahead and start dissolving our sodium bicarbonate into our distilled water. To do that, you'll just drop it in and then we'll go ahead and mix it with our fancy stirring equipment. Once it's visually dissolved, it's good to go. So for this next part, we are going to add our effervescent tablets into our apparatus. And like before, we're going to measure pH every 30 seconds. So get everything ready. Your air stone into the water, your effervescent tablet in hand, and let's get started. So with our distilled water, we're starting at about 8.1. And interestingly, after about 30 seconds or so, we are still at 8.1. So you can really see the power of that buffering capacity of sodium bicarbonate to really hold steady the pH. It's not allowing CO2 to change the pH. So same thing as before, we'll let it go for about a minute and then we will remove our air stone and we will continue to measure. And guess what? We're still at 8.1 in our distilled water. Now there's a little bit change. We're at a whole whopping 8.05. So a little bit of a change, but not really considered significant. And we're staying there until that about 2-minute mark or so. So overall, there was some change, right? But the change is relatively minor when we compare it to our distilled water that we had up here without the bicarbonate. So now let's do the final part of our experiment, and that is to do the same thing. We're going to take now our salt water, and we are going to dissolve our sodium bicarbonate in it, and then we will add our CO2. It's interesting because the salt water looks a little bit cloudy because it already has so much salt dissolved in it. So it makes dissolving that sodium bicarbonate a little bit harder. So now that it's nice and dissolved, we get all our equipment ready. We set up our apparatus. So once everything is ready, we'll add our effervescent tablet. We'll hit start and start measuring. Same thing, we're about 8.4 here. And at 30 seconds will pass, we're staying at 8.4. And after a minute, we're still there. And at 90 seconds, we're still at 8.4. And after a minute, that pH has not changed. So we stayed at 8.4 throughout the entirety of our salt water with our sodium bicarbonate. And so comparing to this one, yes, we both start at 8.4. But you can see how without the sodium bicarbonate, the pH changed a little bit. So our ocean water became acidified, and with our sodium bicarbonate, that really didn't happen. I mean, the change here was minor, but a change nonetheless, right? Versus this one that didn't change at all. And that was kind of the same thing that happened with our distilled water, which shows the power of sodium bicarbonate and preventing changes in pH. So after looking at these data, we can see that the largest drop happened in plain distilled water with CO2. No salts, no buffer, maximum acidification. The smallest drop happens in seawater with sodium bicarbonate. In fact, there's no change at all that happened, and that's because the sodium bicarbonate binds the hydrogen ions that are created when the CO2 reacts with water. This experiment reveals a crucial point. While we often focus on ocean acidification, we can see that all bodies of water, regardless of salt or fresh, can be affected by increased atmospheric carbon dioxide. It's also important to remember that while pH of oceans is decreasing, it's not predicted to become acidic for a while. That being said, the ocean is particularly vulnerable because of all the organisms, such as coral and clams, that live in the ocean and that rely on carbonate to survive. Our experiment also showed that more carbonate ions can slow ocean acidification, but adding it to oceans is really an unrealistic solution. Imagine having to weigh all of that sodium bicarbonate. Maybe tens of millions of tons would be needed to prevent oceanification. Therefore, the only lasting fix is reducing CO2 emissions. The health of our oceans and everything that depends on them is at stake. This experiment reveals how a little bit of science can give us a clear picture of oceans' health and conditions. So I encourage you to keep that spirit of inquiry alive, look at the world around you with fresh eyes, and never hesitate to ask why or how something works, because every question is a doorway to new understanding. Lessons like these are what you'll encounter in your APES class. There's plenty more in our APES course too. Use it as a companion for your class to prepare for quizzes and tests and to get ahead of your peers for the exam prep. Thank you for joining us today. If this video sparked your curiosity, please consider liking, sharing, and subscribing. Your support really helps us bring more science to life. Until next time, stay curious and keep exploring.